Okay, let's get started. Good morning. Welcome. I hope you're all well, getting vaccinated. I'm Karen Aronson, president of the class of 1970. We're thrilled to have you join us. As I was mentioning, this series of environmental talks grew out of our 50th reunion that didn't take place because of the virus. And we decided to take the panel and ask the panelists to speak one a month. And today we have Don Anderson. I think most of you are already muted. Thank you. We will be recording the session and we will be posting it on the class of 1970 YouTube channel. Jesse Hines posts that, and he has or will be putting the URL into the chat box. Thank you, Jesse.
Already done.
Thank you, sir. Or you can just Google it. It's there, it's public, and there are 4 previous talks on tornadoes and invasive species, paradise, and dust in the California air. Our next talk will be on a Monday, not a Wednesday, April 12th at 11 a.m. And Bob Gerber, class of 70, will talk about shoreline mapping, and he will describe how he has ended up challenging FEMA numerous times on their flood maps in Maine and in other states. He'll also talk about the behind the scenes political maneuvering that it took to get FEMA's attention and the social justice issues involved. We'll send out notes about that closer to the time. Today, Don Anderson, a senior scientist in the biology department at Woods Hole Oceanographic Institution, will be speaking. He earned bachelor's, master's, and doctoral degrees from MIT in mechanical and civil engineering. He's going to discuss harmful algal blooms. How do you pronounce it?
Algal.
Algal blooms, which are commonly referred to as red tides. They take many forms. They affect many places all around the world. They poison shellfish, kill fish, contaminate drinking water, cause lots of other problems. He's traveled the world. He's testified in Congress, written lots of books and papers. And today he'll talk to us. He'll speak for about 35 minutes, and then we'll open the floor to questions and comments. If you have a question or comment, please indicate that in the chat function. You don't have to put the whole question or comment there, just say so, or you can. We'll try to go in the order that people sign up. I think it's probably better to wait unless Don would like to take questions along the way, but otherwise, let's wait till the end. And we'll try to go in order that people sign up and let people who haven't had a turn speak before people who've already spoken. So, Don, your turn. Thanks very much.
All right, great. Thank you, Karen. Let me open this up. All right. Well, the topic, as Karen said, is what many people know as red tides, but scientists are calling harmful algal blooms, or HABs. And it's maybe a far cry from what you'd imagine someone with engineering degrees would work on, but it's a real, I consider my career a testimony to the MIT education that really taught us how to think and how to be creative in many different ways. And so now I'm heavily into biology, but in many cases a very a quantitative approach to biology. But in any case, I'm hoping we can explain some things that many of you might have experienced or heard about here or there, but now hopefully can learn a lot more about it. And to get to the very beginning, let's start on land. If you look at this beautiful picture, you see a lot of different flowers blooming there. You might say, oh, it's a nice picture, but if you were a botanist, you'd look at this and see a lot more going on basically under the surface and around this scene. You could actually say there's warfare going on here, that these plants they're blooming. Some of them are blooming actively, some are dying off, but many are fighting for space, they're fighting for water, fighting for nutrients. Some are producing chemicals to both ward off predators, and some are producing chemicals that will ward off the co-occurring plants. And so it's really a dynamic environment that you may not really think of the first time you look at it. Well, that's the way I look at the ocean. This is a picture, a microscopic image of some of the plankton in the ocean, the single-celled organisms, plants and animals. And you see a number of both here. And the ocean is filled with tens of thousands of species of algae. And there's a number of them here. These little golden balls are the ones I work on a lot, but there's a number of different plant species here, as well as a few of the animals that would try to eat them. And it's that diversity. Many of these are hugely valuable and important to the ocean, to the atmosphere we breathe, and so forth. But among those thousands of species are a few hundred that cause harm in a number of ways. And I'm going to go through and show you a lot of examples of that. But it's important to understand a little bit of the biology. These are single-celled microscopic plants, as I said, at least some of the ones I'll talk about. And when they divide, they don't grow huge like a tree. They'll get a little bit bigger, but then they divide. One cell makes two, two makes four, and so forth. And as they continue to multiply, they can actually then change the color of the water, sometimes red, sometimes brown, green. They can have small blooms, as we call them. Remember, they are plants, so they bloom. They can be small like this one in Norway. They can be a little bit bigger like this one in the Chesapeake, the Potomac River. They can be enormous. Here's one that is stretching from Korea to Japan and is visible from space. Now, here's one from New Zealand that I showed for a particular reason. It's spectacular, first of all. That's not false color. That really does look like either tomato soup or blood. But the organism here is non-toxic. It doesn't really produce any harmful compounds, but it still can be very dangerous, because when all that biomass, as we call it, washes into these kind of shallow waters, and that bloom starts to die and decay and run out of nutrients, it then uses up oxygen. And that then indiscriminately kills everything in those shallow waters, top to bottom. And you see this kind of a mortality event. But then let's go down to the Gulf of Mexico. This is an image from near Galveston. And what you see is the discoloration of the water. This is what you'd call as the Florida or the Texas red tide. And if you look closer at that water, excuse me, what you'd probably find would be just many fish that are dead. In this case, though, not dead because of an oxygen problem, but because of a neurotoxin that's produced by the algae that has accumulated and discolored the water here. And those cells are very fragile. They break when they pass over the gills of fish. They release the toxin into the seawater with sea bubbles and such. And then it's not unusual to see scenes like this where you have millions of dead fish and other animals on the beach. These are now from the surface waters. This is not that indiscriminate mortality that goes down to the bottom, although that can happen during some of these events. And I imagine a number of you go to Florida, have been to Florida, may have encountered the Florida red tide. And I'll just give you one example of how severe these events can be. This one was an event that lasted more than a year. It was an event of really national significance. If you look here in this lower left, you see a satellite image of the chlorophyll in the water. And you can see that this particular event stretched from Tampa almost all the way down to the Keys, stretching well offshore. And this is just one snapshot of this event that lasted months and months and months. And the consequences were incredible. Millions of fish dead, hundreds of dolphins, 600 or 700 sea turtles that were dead, hundreds of endangered manatees dead as well. Huge fish like these grouper just killed by these toxins, and in this case, because there was so much biomass, some of the lack of oxygen in the bottom waters. And then another impact from that Florida red tide is that this toxin becomes aerosolized in sea spray. And this is a lifeguard wearing a mask just to avoid that toxin. And if you had a home near the beach, if you were trying to go to a hotel, or if you're on a fishing boat, this toxin in the air was extraordinarily bothersome and caused huge damages. And a lot of people just left the coastline, which of course is why you often go to Florida. Now let's jump to another category. These are the syndromes, the human poisoning syndromes that result when we eat typically shellfish, mussels, clams, scallops that have in turn fed on that algae as food. They're filter feeders. That's how they get their nutrition, and then they accumulate those toxins to very dangerous levels. And several of these are lethal. The PSP problem is one of those. I'll talk to you more about that. That occurs up here in New England, and I've worked on it a lot. Here's a very interesting one. We've only learned about it in the past 30 years or so, called amnesic shellfish poisoning, or ASP. That one can be lethal, but those who survive after just eating a plate of mussels often have permanent brain damage. This one basically is a neurotoxin, again, attacks the hippocampus, the area where we store short-term memory. So if you get badly poisoned, you would have permanent short-term memory loss. And then there are these other syndromes that I won't go through. And then you might think, well, let me go down to the tropics and have a beautiful vacation, and I don't have anything to worry about down there. But I'm afraid that's not true at all. There's another syndrome, this one vectored through fish, called Ciguatera fish poisoning, or CFP. The origin, again, are these little cells here. They're dinoflagellates, a class of these phytoplankton, of single-celled algae. In this case, not free swimming, but they live attached to seaweeds, as you see here. And when they're on the seaweeds, those seaweeds get grazed upon by the little herbivores, the rabbits or whatever of the ocean. And then those herbivores are eaten by the carnivores, and the carnivores are eaten by the bigger carnivores, until you get to a situation where you have, and this is a fat soluble toxin that is biomagnified through the food chain, a lot like the pollutants like DDT or PCBs that you've heard about. So that Ciguatera problem is the most frequently reported marine toxin disease in the world. So not just among HABs, but all other marine toxin diseases. At least 50,000 people a year are poisoned by it, many more probably because it is often unreported. And it is one of these that I honestly go down to the tropics and do not eat any of the coral reef fish like grouper or jacks unless I know where it came from. So, you might be on a boat or on a restaurant and see a beautiful fish like this, and you couldn't pay me to eat that fish in certain islands and certain countries, and just believe me, the symptoms, I've got friends who got poisoned 10 years ago and are still having lots of neurological problems from that poisoning. All right, and if you're in an inland state, you might be thinking, oh, it's nothing but an oceanic problem, but it's not true at all. Let me give you a few examples of what's going on in freshwater. Let's start here in the middle of Florida in Lake Okeechobee. We're going to go back to that same time frame that they think about. There's a red tide going on the west side of Florida in the same time frame. And if you looked from satellites at Lake Okeechobee, what you see now is the chlorophyll from the plants that are in the water. And you see the time series of the bloom developing in just a relatively, few weeks. And Lake Okeechobee is used. They have these big dams and water controls that they have to release water. So even though they knew that there was a very toxic and heavy bloom in the lake, they had to release it to all the canals and rivers. And if you know the geography, you realize that the two main drainages from the lake are to the west in the Caloosahatchee River towards Fort Myers, and then along the St. Lucie Canal off towards the Indian River Lagoon and the inland waterway over here. And what happened in those times then were incredibly, the green slime, the pond scum that you all hear about it in all of these rivers and canals, and just tremendously damaging. This was a toxic organism that produces a very potent liver toxin. It can also cause a lot of biomass problems and oxygen issues. So there were dead fish everywhere. And again, the manatees getting killed once again, this time when they're venturing up towards the brackish or freshwater. And this was such a big problem that, as you see in these political cartoons and this sign from protesters back at that point, that was the election when Rick Scott, the governor, was running for the Senate, and he was being blamed for a lot of this because a lot of the policies that he had instituted got rid of monitoring, got rid of a lot of the water quality regulations and so forth. And so here's Florida, where you have a red tide on this west coast and the green tide basically coming through and hitting both coasts. And this is one of those rare times when in the battle for science funding, it was easy because basically he wanted to be seen to do something about this problem. And I was given a call, is there something we can do? And I talked to them a bit, and honestly, within a week, there was $1 million coming to me to put a team of people together. And I'll talk a little bit later at the end, if we have time, about efforts we are undertaking to try to control these outbreaks, actually physically intervene in the blooms. So another freshwater concern is drinking water. Some of you may remember a few years back, this is Lake Erie. And in this western basin of Lake Erie, here is another bloom of that same freshwater organism called microcystis. And it shut down the Toledo water supply for four days or so, so that 500,000 people were without regular drinking water. And similar events like these have happened in Salem, Oregon, and many other cities as well. So these problems can affect drinking water as well. Now, to further accentuate the diversity of these problems, let's take you to an area affected, and the US is in this category too, but the pictures I'm going to show you are truly spectacular. And so we're going to China. We're going to Qingdao, China. You may know it either from the beer or if you've ever been there. It's a very beautiful city, well known for its beaches. And if you've ever seen a Chinese beach on a hot summer day, this is what they can look like. And
hello, Raymond Farrar.
This is that same beach, though, in 2008.
Fine, how are you?
And what you're seeing is a bloom of seaweed now that is washed onto this beach, very thick, very heavy. And the reason it was significant at the time was that was the sailing venue for the 2008 Olympics. And so these are, the US surfboarder and some of the sailors from other countries trying to practice ahead of time in these incredibly thick waters with the seaweed. So China did what China does well. They called out their people and their military, and 10,000 army soldiers were brought in to move this material off the beaches. Citizens all lined up and had these brigades to move this material, heavy equipment. Over 1 million tons of algae was removed just from those beaches, that city's beaches and the bay there. And they trucked it inland and buried it. And they actually were able to hold the Olympics in fairly clean water. But these are just some pictures of the incredible extent, almost biblical in proportions. People still went in the water. They still had tried to have fun. And you may wonder how did this happen? And if I were talking to an audience right in front of me, I'd say, how many of you like and eat sushi? And quite a few of you would raise your hands, and I'd say, well, you're actually indirectly responsible for this, because further south in China is a huge, massive area where they have aquaculture facilities for this seaweed called Porphyra, which we know as Nori. That's the crispy brown kind of dried seaweed that you get with sushi. And that's what's growing on these nets that you can see here as far as the eye can see here, with the tide being out, here the tide is in. But notice on the ropes and the bamboo around this, that is this contaminant alga called ulva, and that is the green from the green tide. And what the farmers do is they clean that off, they throw it onto the mud, the tide comes in, picks it up, it aggregates. The water is very nutrient rich, it's polluted in many ways, and these mats form that then continue to grow as they're being moved to the point then where you can see them from space. They are that big and that extensive. It's extraordinary area coverage. And then of course the wind blows, and here's Qingdao, and this whole Shandong Peninsula then can get covered with this seaweed that all started from that aquaculture area, which is down here in Jiangsu province. And just to show you that was 2008, and you saw how bad it was, but it has continued every year since then. I don't have the most recent data, but it has continued, covering massive areas of the ocean and the coastline. And if you wonder, I mean, am I really right about this being something having to do with the Nori aquaculture? Here's a plot of the amount of hectares covered by that aquaculture industry. And you can see that right around 2008, 2009, there was this huge increase. And I should say that this green tide was happening before. 2008 brought it to everyone's attention because of the Olympics, but it's also clearly gotten worse. So those are just some of the examples of, and there's more, of the tremendous diversity. And that's why we have this general term of harmful algal bloom and not red tide. So what kind of trends are we looking at? Well, let's first look to the US. Here's a plot I produced years ago, actually for Congress. And this is what we knew back, actually, this was right when I was beginning to do my thesis. What did we know about these problems in the US? You can see very few dots and triangles and whatever. And this is the situation now. You can see areas where the problem has expanded, like in New England or here into Puget Sound. You have expansion down here. You have new syndromes entirely. We didn't even know about the ASP syndrome. And so there has obviously been a huge expansion of these problems in the US. And if we look globally, and this is just for paralytic shellfish poisoning, this is what we knew back in the 70s. This is what we know now. And, you know, I could go into lots of, well, I'll hold on, I'm getting ahead of myself. I wanted to also try to show you some freshwater trends. And this is actually, these are data, these are hard to come by in terms of how often these events occur. And someone cleverly thought, well, let's look at all the news reports where they mention these blooms and the cyanobacteria, that the group of organisms causing these. And so these are newspaper reports of these events in 2010, 2012, and 2014. These are cumulative, 2016, 2018, and here's 2020. And if you toggle back and forth, this was basically what was known. The orange is just 1 year's reports of these events in the US. So you can see that the freshwater problem is just significant and growing as well. So I'm sure you're wondering why. What's going on? The scientists have. We all are in agreement that the problem is getting worse, but it's for a lot of different reasons, and some of it's natural, just natural species dispersal by just natural currents or storms. I'm going to talk to you in a bit about cysts. These are resting stages, like seeds, and it's easy to move these around. There certainly are more people like me, my colleagues. We have better chemistry, we have much better communication. We are finding problems that always were there, but we didn't know about them. There's increased aquaculture where we have more resources and more monitoring. Human activities, whether it's ballast water, we're carrying them around the world. And of course, eutrophication. These are plants. A lot of the pollution that goes on in the freshwater and the ocean is a source of phosphorus and nitrogen and other elements that are needed for these little plants to grow. And finally, climate change and global warming is playing a role here. And I wanted to sort of piggyback onto the talk that John Eichelberg gave us about what's going on up in Alaska with climate change. And I brought this slide back again to show you that my eye, and this of course there's lots of red dots, but it goes to where there aren't red dots. And notice we don't really know much about this PSP problem up here in the Alaskan Arctic, or even over here in the Soviet Arctic, and up in Greenland and in other areas. And I've been very interested in trying to get funding to go up there for years and years, and a lot of times just was very difficult to get that support. And the argument that I was giving was, look, everything's warming up there very fast. Doesn't matter what you look at. If you look at the annual temperatures, if you look at the sea ice extent, which is decreasing, if you look at when the Beaufort and Chukchi seas freeze up, you can see it changes from November to December. It's getting later and later in the year. Well, why is that? If you're an oceanographer like me, you look at the water, and you can see here in this simple diagram that of the energy from the sun that hits typically the ice, 85% of it is reflected, and only 15% of that energy is absorbed. But if that water then, if that ice melts and you're looking at the water, it's the other way around. In this case, 93% of the energy goes into the water and only 7% is reflected. And so that's one of the reasons that the waters up in the Arctic are warming so rapidly. And it's one of the reasons that people are saying that's one area of the world that is warming faster than any of the other areas. So it's an area that I've wanted to just go up there and see, are these organisms there? Are they entering that region? Have they always been there? Just what's going on. So we've been running large cruises on vessels like this. This is the Coast Guard icebreaker Healy. We've used this a number of occasions, running these transects through the Bering Strait, up here into the Chukchi Sea, and over towards the Beaufort Sea. We've been collecting water samples in the surface, sediment samples as well. And the reason we're collecting sediment samples has to do with the life history of these organisms. Here's this Alexandrium catenella. That's one of the PSP organisms. It has this cyst stage that's in the sediment, and it germinates certain times of the year to produce these cells. We call them vegetative cells that are photosynthetic. They swim, they aggregate, they divide, cause a lot of the problems with toxicity. But there's this, then they go through a stage where they make gametes that fuse together to make a zygote and produce these cysts. And actually, my thesis work was involved, we discovered these cysts years ago and realized how important they are in the dynamics. And I still continue to study them. So one of the first things you do if you finally do get funding to go up there is to map out the cysts in the bottom sediments. And this is a map from 2018 and 2019. The colors show you the abundance of these cysts per cubic centimeter in the sediment. And there's just several things to point out. The first is that there is this huge seed bed, we would call it. Remember, these are like the seeds that are going to germinate and start a bloom. This huge seed bed here in this area near what's called Ledyard Bay and the Chukchi Sea. Notice this line. We can't go over here because it's Russian water. So this even clearly extends much further than we have been able to resolve here. And the numbers that we're seeing, first of all, it's a massive seedbed in area, but the numbers of cysts we're seeing are the highest ever reported globally. I've done this work all over the world. This dwarfs the other areas where we see this organism. And then if you look up into the water column, now we're looking at these cells in these same cruises. Now you see this bloom sitting right above that seed bed. And again, with cell concentrations well above the levels that we know cause dangerous toxicity in other regions of the world. And so together, the location of the cysts, the location of the blooms are telling us that it looks like the blooms in that region are going to be what we'd say locally originating. In other words, the cysts germinate to inoculate the water, and then they're going to bloom there. And then they're going to self-seed. They're going to drop their cysts in that same area. And that means you're going to have this happening year after year after year. It's what we've seen in many other parts of the world. And therefore, this is very significant implications for the subsistence food supplies of all the indigenous communities up here. They eat everything. They eat birds, seabirds, walruses, whales, and we don't know anything. And that's what we're just starting to get a handle on. How dangerous is it for them? What are the vectors for this toxin into these different food sources? And what are the sensitivities of those food sources to these toxins? And just to show you that climate change is involved here, this is just 20 years worth of bottom temperatures in Ledyard Bay. And what you see is that in about these first 10 years, starting in 1999, the water's right around 4 degrees Celsius, but it has warmed about 3 degrees since. And that may not sound like a big deal, but at these temperatures, the cysts of Alexandrium can take 2 to 3 months to germinate. And it basically precludes a bloom developing because it takes so long and such a slow process. But out here, it just takes a week to 10 days for those cysts to germinate. And you can really have that huge number of cysts, this sleeping giant, as we've been calling it, awakening and inoculating those waters. And the final comparison here is I've done an awful lot of studies here in the Gulf of Maine with the Alexandrium. And then here we've got on this exactly same area, not only spatially, but the same scales for the cyst abundance. And what you see is that this Alaskan Arctic cyst seedbed is a good 5 times larger than the one over here in the Gulf of Maine. It has a good 15 times as many cysts in it. And yet this one, which looks rather wimpy now in comparison, it has been fully capable of causing annually recurrent, very large scale blooms of this organism. And here's just one example of a bloom that we ran all these cruise transects here. Look at the size of this bloom in the Gulf of Maine, all initiated from this seedbed. Imagine what we can expect from what we've just been discovering in Alaska. So anyway, to move on, I want to try to cover some of the new technologies that we're using to try to deal with these. And they include forecasting, new sensors for enumeration and toxin detection, and as well as, if we can get to it, some bloom control technologies. So starting with the models, there's several types. The empirical or statistical types are just where you collect a huge amount of data, the cells, the environment, and then you try to find correlations so that you can then say, with this set of conditions, we will have a probability that this bloom will or will not occur. I find these others much more useful and much more challenging. There's 2 types that are numerical models. To talk about those, I wanted to point out, though, these are weather forecast models. You've all seen them, you all benefit from them. They use the same physics that we do to look at the movement of the ocean. They're both fluids, but I actually think that the weather forecasters actually have it easy, because all they're really trying to do is look at the properties of that water that they're able to model. They're not having to tell you whether it's birds or gypsy moths or something else, some biology, how many of these gypsy moth, let's say, are being carried by this storm as it moves up in this way, how many are germinating, how many are hatching. So when you throw in biology, the forecasting gets really difficult. So one way people get around it is to ignore the biology. Here's that Lake Erie situation. This is this type of approach, which you would call particle tracking, basically is used for blooms that are visible from space, typically. The ones in Florida, the ones that in freshwater systems like this. You can take this visible image, you can look at the chlorophyll signature of that population, and you can see this. And then now if we just look at this eastern basin, you can actually, here's the chlorophyll, you can digitize it into particles and then propagate those particles forward just like you would an oil spill or something else with a hydrodynamic model, and then turn it back into chlorophyll in organisms, and then be able to alert water supplies and so forth about pending danger. So, but in this case, there's no mortality, there's no growth, it's just passive particles. If you try to get to areas though where we can't see these blooms from space, you then it's a lot more difficult. You start to have to develop population dynamics models. And I have to do this really quickly, but to do that, what you do is you start with a hydrodynamic model. This is the one we have for the Gulf of Maine region, very well resolved, very good resolution of the physics, to which we then have to add a biological submodel. We start with a map of the cysts in the bottom sediments, just like the ones I was showing you earlier. This tells us what time of year, for these are lab studies, these cysts will germinate, how fast they germinate as a function of temperature and light. And then once they've germinated, how fast the cells grow as a function of salinity and temperature, nutrients, and so forth. And you put all that together, you couple them together, and you get a simulation like this, starting from the cyst. You can see the dates here. This is April and May. See this bloom moving down the coast of New England, out onto George's Bank, and so forth. And this is being used tremendously. Well, it's very useful for forecasts, for looking backwards. But one of our problems is, you can see we started early in the year, and once it moves ahead, we have no easy way to correct for things that have gone wrong. So if we think about how can we improve our HAB forecasts, one of the ways to do it is to put out instruments, what we'd call in situ sensors for cells and toxins, that are going to give us this real-time data that we can assimilate into those models and improve forecast accuracy. And the best example of how this works is again, back to the weather.
Right.
We have such great weather.
Yeah, so
forecasts.
hold on 1 second while I turn.
Okay, so we have such great forecasts because there are these meteorological arrays of instruments on land. We have weather balloons and so forth. They collect data that is fed back to, assimilated into those numerical models that are then run again and forward, and you keep having these corrections. But we don't have those instrument arrays yet in the ocean for HABs or lots of others, although we're building them rapidly. And that's a lot of the energy in my lab these days, is using these instruments. We call them autonomous, robotic, in situ HAB sensors. One's called the ESP, and I'll explain it, and the IFCB is the other. So the ESP is called the Environmental Sample Processor. It's this little small trash can size instrument here in this array that goes into the water. And you can see the various surface moorings here. This takes a lot of ship time and everything to put these in the water. These are complicated instruments, but what they are able to do totally robotically is to look at the species that are there, as well as the toxins that are there, using DNA technology here, using antibody protein technology here. And I'm going to show you a video. I hope it streams well to all of you, and I'll sort of be quiet for a moment.
Life, and the search for it, is perhaps our greatest adventure. On Earth, the most abundant forms of life are those that we cannot see with the naked eye. 3 kilometers below the ocean's surface, microscopic life forms flourish in the liquids seeping from the Earth's interior. What are these organisms? How did they get here? And what impact do they have on our environment? Each year, massive blooms of algae render parts of our ocean toxic to humans and wildlife. Imagine if we had an automated detection network warning us when harmful microbes were present. As we further industrialize the ocean, what is the effect of microorganisms in our harbors and off our coasts? Is there a better way to monitor how we are impacting the planet? Fish farms are especially vulnerable to outbreaks of harmful algae and disease. What if we were instantly notified of changes to the health of the waters around aquaculture areas? The Environmental Sample Processor is an automated laboratory able to detect microorganisms in real time. Consider the mysteries this biological and chemical sensor is beginning to address. Our ocean houses life forms we know almost nothing about. Where do they travel? How is humankind impacting their environment? And how are they, in return, impacting ours? The ESP provides us with a model for how we might monitor the health of our ocean in the future and seek out new forms of life in the darkest depths of our world and in the farthest reaches of others.
Okay, well, the reason I showed that was in part because if you noticed at the end, some of the work on that development of that instrument was paid for by NASA. Why would NASA care about the ocean and what's going on at the bottom and about HABs? Well, it cares much more about what you already know. This is the Perseverance rover that is on Mars, and there is not an ESP on there, but the plan is to be able to take that technology as it gets smaller, as it gets much more miniaturized, and to use it. And think about what's having to happen with Perseverance right now. They're taking samples, looking for life, and they have to leave them on the surface of Mars for 10 years or so until some other craft can come and pick it up and bring it home. And what they really want is to be able to do the analysis right there, which is what we are doing. So they are taking an instrument that was developed for the use in my field to look at these harmful organisms and to look at the toxins, the molecules they produce, and then are trying to adapt this for use in the search for life on other planets. And someday we can do it in real time almost and not have to wait 10 years. All right, the IFCB is another very powerful instrument. We're using it heavily. It's basically an automated submersible microscope that uses imaging flowcytometry, you know what that means, to find particles that have chlorophyll in them and take high resolution images. 8 to 10 in a second, hundreds of thousands of them in a day. Here's a picture of a dashboard of just a few minutes worth of sampling in an area. These are all actually Alexandrium cells in their different life history stages. And so much information is collected that humans can't deal with it. So we have to use the machine learning and the neural networks, much like the facial recognition software that you hear about these days, in our case, to identify and to count the cells. So when you put these together, one of our visions then for the future is to have an array of these ESPs and IFCBs along the coast that are giving us early warning and what we call situational awareness, but which is also providing data that's going to be assimilated into those forecast models. And to show you some of the more recent developments, right now, the third generation of the ESP is shown here. And it's now up in this end of this autonomous underwater vehicle, or AUV, and it's about the size of a soccer ball, and it's using microfluidics like this. So you can see it here in the water, sending its information back. The IFCB is a much more energy hungry device because it has a laser running all the time. So we've been putting it now on these autonomous surface vehicles like this one, which you see have solar power. And there's the IFCB underneath. And one of the things that I think is just marvelous about these are that they can do what we call station keeping, where you just basically say, stay right here. And even in winds up to 45 kilometers per hour or waves that are 15 meters high, this can sit there just like you would have a mooring, but at much, much lower cost. You can also tell it, please go and run this transect and then come back, and it'll run a transect collecting samples, giving us all this great information. So that vision that I was showing you is now being improved, where we have fixed instruments as well as these mobile assets, giving us much broader view of what is happening. So to wrap up, I just have a couple minutes more that I wanted to talk about what's in, I'm sure, a lot of your minds. What can we do about these? Can we actually control them? And it's just such a difficult problem. It's nothing like what you're able to do. I use this example here where you can put down a chemical that only kills crabgrass or only kills weeds. So it's killing a certain group of plants, but letting other plants grow. We are nowhere near the stage of having that capability where we can target these HAB species and leave all the other organisms in the ocean unaffected. And so the chemical control is just, it's very much in its infancy. There are viruses and bacteria that can control these organisms as well. Some of them have exquisite sensitivity towards the species we want, but then there's all these environmental concerns about will environmental groups let us release these into the ocean? And how do you have enough of them ready when a bloom occurs and so forth? So believe it or not, physical control is turning into the approach that is used the most, and it's almost archaic the way it's being used. The most practical and acceptable large-scale bloom control strategy being used worldwide is literally spraying solutions of clay onto the surface of the ocean and relying on flocculation to remove cells and in fact, toxins. The way that works is that the clay particles are actually treated with a polymer called polyaluminum chloride that makes the clay a positive surface charge. So the algae have a negative surface charge, and that there is then this attraction, and you can flocculate and aggregate the red tide or the HAB cells together with the clay particles. But also the polymer makes these chains that bridge between the particles. And as this material gets heavy and falls, it basically is like a net that captures more cells as it falls. And as a result, you can use very tiny amounts of clay, literally a packet or two of equivalent of sugar packets would be enough to treat a square meter of the water column. So it removes a lot of concerns about environmental impacts. So why is clay being used? Why is it popular? Well, it's been used for more than 20 years in Korea and China on a very large scale. It's abundant, it's cheap, it's present everywhere. Every time it rains, the soil, the cloudiness in rivers, much of that is clay washing into the ocean or into lakes. And therefore organisms are very used to it. It has very low environmental impacts. So here you see Korea has 7 boats like this, huge boats that will spray this clay around these their fish farms and prevent damage to the fish farms. China has a fleet of these boats also that are spraying and treating all of these outbreaks. Now in the US, we're nowhere near that. We have such strict environmental concerns that we have to start small. And with that money that I mentioned from the state of Florida, we now have 6 or 7 permitted sites along the west coast of Florida, typically canals like this one, where we can then do these kinds of experiments, put a boom across there, measure parameters, mix the clay in, spray it from land in this case to remove the cells and do all the analyses and so forth. And we're making progress, but we're way behind other countries in that regard. And the dream we have, or the vision we have, is to move from this type of testing to more large-scale treatment to eventually the kind of things you see for either mosquitoes or, let's say, forest fires. And so to finish up, just a couple thoughts. I hope I've shown you that this HAB problem is probably much more serious and much more of a threat than you thought. It's threatening public health, fisheries, aquaculture, tourism, everything, water security in both freshwater and marine systems. It's clear that the problem is going to continue to expand because of our population growth and pollution and even climate change. And right now we have many regions that are threatened by multiple HAB species and toxins. They occur at different times, different habitats within a region. And so the managers that have to protect public health and resources are really struggling. And that's why a lot of technology is needed, both these in situ sensors, the modeling that we're doing, and ultimately these bloom control strategies. So I want to make sure that a lot of the agencies and groups that have supported this are acknowledged, but I'm happy to stop and take questions.
Thank you, Don. That was fascinating. The first time I came across red tides was actually when I was at MIT in Boston. Let me ask one question and encourage people to post in the chat if they want to ask questions. I see at least 2. What about, you mentioned when Lake Erie had a problem that there was a drinking water problem in at least one city. Have there been growing numbers of drinking water problems because of algae issues? And
absolutely. The The other case I mentioned that was prominent in terms of size was in Salem, Oregon, again, where several 100,000 people were out of couldn't drink their tap water. But virtually any lake or reservoir that's used for drinking water supply has issues like this, and they are trying to remove these populations, sometimes with copper sulfate and other treatments, but what that does is release these toxins into the water anyway, and then you need activated charcoal or something else to remove it. And so it's a constant battle for these water treatment all over the country. And so yes, there are occasional shutdowns, some big ones, but think of it as an ongoing battle where it's getting worse and worse. And these utilities are asking for help from remote sensing or whatever to be able to see these blooms and give them some advance warning, because these treatments are expensive. So yeah, the drinking water, I would say that, in fact, I give advice to young graduate students all the time, and I say, boy, if I'd go into the freshwater side of this field more than the marine side these days, because that is growing the fastest. That's where the demand, the problems is growing the fastest.
Well, I wasn't so aware of it. Thank you so much. And I think you should not retire. We need you. Peter or Siva Kramer, unmute. You have a question.
Unmute. What made the 2017-2019 Florida Gulf red tide disappear? Was it a natural, I mean, did it just burn itself out, or is it still festering?
Well, it's a bit of, well, it's both actually. And remember, this one lasted, I don't know what it is, 19 months, some 17 months, some huge amount of time. So when you say it disappeared, for the longest time, it just kept persisting. And what happens is these cells grow and then they're taking up nutrients. And a lot of people in Florida blame the sugar industry and big agriculture and all the fertilizers being used and going into that Lake Okeechobee and the Caloosahatchee and coming out into the coastal waters. So their argument, those who were saying that the bloom was fueled by nutrients from land, argue that's what kept it going, this additional human input. But what's also happening is cells are dying, they're breaking apart, the nutrients are getting remineralized and coming back, and new cells then are able to use them, and that bloom can persist. And that cycle obviously can continue for a long, long time. But eventually, whether it's a predator that comes in, a virus that can bring that bloom down, it's actually not known what caused that bloom or what causes that bloom to really terminate. There was a very recent funding of I don't know, about a $5 million project awarded to my colleagues in Florida to address just that question. Why do these blooms end down there? But they do ultimately end, again, whether it's because there are predators and viruses or they run out of nutrients. But when they end, that organism in those nice warm tropical waters doesn't just disappear. It hangs around, like you were asking. If you go out into the middle of the Gulf of Mexico, you can always find these cells of what's called Karenia brevis. And then it's just waiting for the conditions to bloom again. And so it will happen. I mean, I expect last year it didn't happen much. I expect this year we're probably likely to get more Florida red tides, and we're going to go after it with some of these control methods. So I'm actually hoping some red tide happens this fall and winter, but it will happen from these cells that are just hanging around, survivors from the past blooms that then get these conditions and flourish.
Thanks. Mike Prager, unmute, your turn.
Thank you. Don, I was wondering if it's been possible to disentangle the effects of nutrient enrichment from runoff and other sources and warming. So it seems to me both of them could really act together. So what's your insight to that?
Well, first on the nutrients, I mean, I've already told you there's a big controversy down in Florida among a lot of people who think the real root cause are the nutrients from land. But that organism grows much of the time out in the middle of the Gulf of Mexico, which we think of as a biological desert. It's oligotrophic. It's not nutrient rich. So that's sort of like a cactus. It doesn't need a lot of water. So if you give it a lot of water, are you really helping it? And so there's this constant argument going on between some scientists and one side and other scientists, and the public's in there. So the Florida story and the story for many other these HABs, the link to nutrients is it's there in some areas and not in others. In the Gulf of Maine, I don't blame nutrients from humans at all. 95% of the nutrients of our blooms use are coming from natural water masses, natural sources. But other areas, it's much more, a much bigger linkage. Freshwater, the story is much clearer. Lakes, reservoirs, rivers, those blue-green algal blooms really are thriving on excess nutrients from land, from fertilizers, from sewage plants. That one is really clear. Almost no one will argue. So a good way to try to reduce those are very expensive and taking a lot of time to do watershed control and nutrient controls. All right. But your final question was trying to tease apart nutrient effects from climate or warming. That's easier to do in areas like Alaska, where we don't have pollution, don't have nutrients. We can really see a real climate story, but much more difficult, like with the freshwater. And there, unfortunately, there's a double whammy. One of my colleagues wrote a paper in Nature that basically says cyanos like it hot. That's referring to cyanobacteria that cause those a lot of those green blooms. And they really like it like warm water. And as the warmer the water gets, the better that group of organisms does, and the worse that other competing and non-toxic algae will do. So there you get both nutrient effects and warming combining. And that's one of the reasons I see the freshwater HAB problems being the real growth area, if you will, for the future.
Let's hope not.
Yeah.
Thank you.
Thanks. Tony Picardi, did you want to make some comments or?
Yeah, I was interested in the blooms of filamentous green algae. I have a frog pond and a larger several acre fish pond. The frog pond I can control with putting in a flocculant. And so the question there is when the algae aggregates sink to the bottom, do they decompose and then add nutrients to the pond afterward? It seems like the larger SAV grows and uses up the nutrients so that the filamentous doesn't come back in the frog pond. In the larger pond, I can't do that. And in the last, there's been a lot more filamentous green algae, and doesn't seem a way to control it there. And there's also pondweed in the last year, which is persistent over the winter. So I'm thinking maybe warmer temperatures are causing green algae blooms that
Okay, there's a number of questions in there. So the first thing is I would expect that what you're actually experiencing is not necessarily green algae, but blue-green algae, but that's just more of a technicality. But the flocculant that you're talking about, yes, it will carry this material to the bottom, and in time, there will be bacteria and other organisms that will break down that biomass and release those nutrients back again. So yes, you would have this cycle that will continue and continue. And in fact, one of the methods that they do try to do, I've been to Lake Taihu in China, where they have these huge pipes that they're basically sucking up the bottom mud and discharging it and carrying it off to land, because there's this legacy nutrient supply. That's what it's called. All of the nitrogen and phosphorus that have come in are in the sediments, and no matter what you might do upstream in the watershed to reduce fertilizer use or whatever, these nutrients are going to keep coming out of those sediments and keep fueling these blooms. So in some cases, I hate to say it, dredging is one of the approaches for like your bigger pond. But another one, it's a little bit expensive. They actually have a material called Phoslock, which is a chemical that when you put it down, it actually caps the bottom sediments in such a way that when the phosphorus that's trying to be regenerated and get into the water column is blocked at the surface of the bottom surface, and that actually works extremely well. But behind your first question, that was one thing I'm glad you raised. One of the problems we face in telling people we want to treat the Florida red tide with these clays is that we will be bringing down not just that biomass, but toxins to the bottom. And the first question people ask is what happens to the crabs and the sea urchins and the coral and so forth. And there's two ways to answer it. The first is the studies we've done show that there is some impact to them because in fact there is a toxin going down there. But what we're telling people is don't judge this treatment against a pristine water body with no red tide where everything's healthy. You've got to compare this treatment to the no treatment alternative, which is already killing everything on the bottom because of either oxygen or toxins that are continually raining down during these long blooms. So yes, you do bring down material, and it will regenerate, but the comparison to the actual bloom, it's much, much more severe. And in fact, if we can stop that bloom, then we won't have one that might last another 5 or 6 months if we can stop it early enough. So there were a lot of things in your question. I hope that covers them.
Thank you. Claude Gersel, unmute.
First, Karen, I wanted to thank you and Don for putting together such an interesting program. Some people have looked at ocean fertilization as a way to sequester carbon in the ocean. So if we did this in open ocean and then added a flocculant to take the algal growth down to the bottom in deeper ocean where it would just sit there, could this be an effective way to sequester carbon in the ocean?
The second part of that question is fascinating. I had never thought about it. I know a lot about the ocean fertilization proposal. For the others, the concept is a little bit nuanced in that there are parts of the world's oceans where we call them high nutrient, low chlorophyll. And that would be like in the Northwest Pacific and in other parts of the equatorial Pacific. And so there's a lot of nutrients there, but there's not the biomass, there's not the algae that we think should be there. And so that means something else is limiting. It's not nitrogen, it's not phosphorus. And it turns out it's iron because it's so far from land. And iron's needed, it's part of the chlorophyll molecule. It's needed in a lot of physiology. And the amazing thing is that if you can supply iron, you can, I forget what the multiplier is, but it's like 500 or so atoms of carbon for every atom of iron you supply. I think it actually may be even bigger than that. But whereas with nitrogen, if I fertilize with nitrogen, I might only get get a few carbon atoms. Okay, so there's a real lot of bang for your buck if you fertilize the ocean with iron. So there've been a number of experiments, honestly, where people have gone out on ships and sprayed iron onto the surface of the ocean and proven that they in fact take up, they get a green spot in the ocean where the algae that are there grow on those excess nutrients, and they were limited by the iron. And so people have proposed that as a way to then remove more CO2 and to scavenge it. But the problem always has been that, remember what I said about the recycling of the nutrients in Florida? That's going to happen again. You get that green spot, everything grows, it gets grazed, it starts falling down, all of the animals are eating it. That material and that carbon doesn't reach the bottom. So that's why the question I think is very clever. I have never thought, and my colleagues have never thought about whether you could do that and then follow up with a flocculation treatment that carries it down to the depths where it's really sequestered and you truly have actually done some carbon removal. Whereas the first scenario I said, you don't really remove that much carbon if it recycles. So that was it. Actually, thank you for the question.
Thanks, Claude. Thanks. Don, Earl with,
Thank you, Don, for an excellent presentation. I'm working on windblown dust control at the Salton Sea, which is hot, or at least warm, very nutrient rich and shrinking in size. And one of the concerns is the viability of cyanotoxins on the dried lake shore being caught up in windblown dust and exposed to people with respiratory problems. What is the
Yeah, there's a lot behind that question. So the first is that yes, many of these toxins are quite viable when they are dried. In fact, many of you may have heard of situations where dogs will die after there's something tasty about this dried algae along the shores of lakes and reservoirs, and we've had it happen right here in Cape Cod where animals have died from eating this dried material, and it's clear that the toxins are still there and still very potent. And so you know, I don't know, but I'm sure people do, how long they are viable. I mean, the Salton Sea, as it dries, there's probably material that's 10 years old that's there, but certainly freshly dried or material that's dried within a year or so can be very dangerous. And then you would worry about it becoming airborne. And one of the growing areas in this freshwater field is how much are people inhaling these cyanobacterial toxins, either from the source, like you've mentioned, the dried material, or from the spray, the wind that's either coming off of lakes and rivers or during recreation. If you're on a boat or you're swimming, how much of it are you inhaling? And to make this even more alarming, if you will, there have been links made between people living near ponds and lakes and rivers and dementia and other cognitive impairment diseases. And people are starting to try to link those to certain of the materials produced by these cyanobacteria, one called BMAA, but some of these other many toxins produced by these organisms may be actually impacting people in an airborne way, in a manner that the toxin in Florida affects people when it's inhaled, but it just makes you get something like the flu and your nose runs and you sneeze and all that. And there's really, the only long-lasting effects seem to be from people who are asthmatic, who have some problem afterwards, but nothing to the extent where you could actually have some neurodegenerative effects. So what you've touched on is a very hot and important topic these days.
thanks. I love these programs, both because of the speakers and because of the questions and the discussion. Ed Pallo, unmute.
Okay, thank you. A very interesting talk. I was struck by your comment, and I've seen it when being down in Florida in the tropics about eating large fish that feed on reef fish, that there's a significant issue and can cause health issues. And you said you wouldn't eat anything unless you knew where it came from, and those kind of things. How do you ensure that the fish is safe?
Yeah, boy. Okay, so this is about the ciguatera fish poisoning, and first of all, certain kinds of fish are fine. I mean, I almost showed it, but I just took it out because of time. There are posters you can find that'll say, never eat these fish, the horse-eye jack, the yellow-eye jack, this group or that group, or these other fish are sort of risky and these others are less risky. So part of it is knowing which fish people are serving. And the further you get away from a reef in the tropics, the safer you are. So if it's a flying fish or if it's a mahi-mahi or something, those are typically going to be safe to eat. It's the ones like the barracuda. You never, ever eat a barracuda, especially a big barracuda. I would not even eat a big barracuda along the Gulf of Mexico coast, but certainly never in the tropics. And So you have to know what kind of fish is being served. But then on islands like St. Bart's or St. Thomas, many of them, one side of the island is safe from ciguatera, and the other side, the leeward and windward side, is very dangerous. So that's what I mean by knowing where the fish came from. There was an interesting situation where, with the hurricanes that swept through the Caribbean, some of the islands got hit really badly, and so the fishermen would then go from one island to another to fish, and they didn't know what the locals did about where to fish and where not, to avoid what they just call fish poisoning. And so a lot of people got poisoned because fishermen were just fishing in areas where the locals knew they shouldn't. So that's another way. Now, you'd love to say, well, why isn't there a little test that we can use to tell us if a fish is safe? And years ago, they made one in Hawaii. It was like a little toothpick that had sort of that white out material on it that you use for typewriters, and you jab that into the fish and it would absorb some fluids, and they would run that through a test. But it cross-reacted with too many things, and it gave everything. It false positive, false negatives, and really just wasn't useful. And that was 10 years ago, and there still is no good test for ciguatera toxins. And a lot of people are trying. It's just a hugely complex molecule, and it's a very difficult challenge. So right now there is no quick test. So the best way to do it is either have local knowledge, like I said, about where the fish came from, what kind of fish it is, or to do like what I often do, which is to eat cod and salmon when you're down on these islands. Hate to say it.
We're supposed to eat more fish to be healthy, and
yeah.
Thanks.
And I should also add, Karen, that I can be one of those restaurant goers who strikes fear into the heart of the maitre d' or the waiter when they say, okay, we're having grouper, and I'll say, well, where's it from? Was this aquacultured? Was it, what kind of grouper was it? And it's like, oh my God, how do we know this? But a little bit of knowledge can be too much sometimes.
Okay, Ray with a question about Guatemala. Unmute.
Unmuted. Okay. Thanks, Don. Very nice talk. So I've done some volunteer work in the Lake Atitlan area in Guatemala, and they have serious problems with HAB and both human and fertilizer runoff. Are you familiar with that? Have you done any work helping with that?
I haven't done work in Guatemala. I'm doing some work in Peru now, and they've tried to get me trained into some work in Lake Titicaca, which has similar issues. These high altitude lakes that are just getting too much pollution, and therefore these blue-green algal blooms are flourishing like it's happening everywhere else in the world. But actually, the work I'm doing in Peru right now is more on the coast, where they have a really growing and very good bay scallop industry in a place called Sechura. And if those of you that like bay scallops, they can be really expensive. But if you look for the ones that come from Peru, they're the same as the ones we get in the US. They're extremely good and a lot cheaper. But that's an industry, like others, that is now having to worry about blooms that can just come in and wipe out a year's harvest. And so they're bringing me and my Chinese colleagues, are the ones that have been working hard on this control method, to see if we can get them set up to actually be ready to intercede in a bloom. But that's on the coast. So no, I haven't. I'm just too busy, really, is what it comes down to. Much as I would like to get involved in some of these beautiful mountain lakes, just can't do it.
Okay, thanks.
Thanks. Peter Dreher, unmute.
Can you hear me now?
Yes, indeed.
Yes.
Okay, great. And Don, you and I have talked before about this ocean iron fertilization. I'm from the Palm Beach Club with Paul, with Dr. Gerstel, Claude, and another one of our members, Sharon Seagren, has a project. She actually lives in the Jupiter outlet or inlet, I guess the inlet, And so her home is right on the beach. And so she goes swimming there every morning. And so she feels the burning sensation from the tide, the red tide, and, is obviously disgusted about it and wants to do something about it. So she's developed this system for harvesting algae from Lake Okeechobee. And I'm being in the toilets fertilizer business in Africa, say, hey, let's turn this algae, which is loaded with phosphorus and nitrogen and potassium. It's a great fertilizer. It also has water, and let's use it to fertilize the strawberry fields of Florida. But now you're telling me it may be loaded with HABs. Lots of, if it's just cyanide, we can cook it off. But apparently some of these things is not just cyanide. And we're going to poison all the strawberries, and it's going to go in the strawberries and kill everybody in America. Is that true or not? Are we smart or stupid trying to harvest algae as a fertilizer out of Lake Okeechobee?
Boy, what a loaded question. I don't want to call you stupid, but it is a real risk. And the reason is just what you've said. Those toxins, by the way, as much as I'm saying these are cyanobacteria, cyanide is not produced by it's not one of the toxins produced. I don't even know where the name cyanide came from. Maybe it's because people that are poisoned turn blue, and that's the color of cyan. But these toxins are called microcystins. There's lots of other cylindrospermopsins and anatoxins. There's a whole suite of them produced by these blue-green algae. And they vary in how long they will remain. But there have been studies that show that if you spray a field with some of these, with water that came from some of these areas, that the plants will have then some of these toxins on them. They are not taking up the toxins. It's not, these are big molecules, and plants don't really have that necessarily those capabilities. They can take in CO2 and create their own proteins and so forth, but they're not going to take in these big molecules and have it get internal. But they will be covered on the outside, whether it's lettuce or something else. So you can really wash things well and get rid of most of it, but there is a risk. There's no question that there is a risk. But one of your questions was, what are these toxins? A lot of them are neurotoxins that have these specific targets. Saxitoxin is the one that we have a lot of. That's the PSP toxin. It's a whole family of compounds, but the parent compound is called saxitoxin, and it blocks the sodium channel. It's like a cork that blocks the sodium channel that allows the flow of sodium in and out of cells. So when you get poisoned, you lose control of your ability to breathe, your diaphragm, and you actually are not able to breathe for yourself, and the therapy would then be something like an iron lung or someone breathing for you. And this group is one of the few that I forgot to say it. I'm just taking this opportunity to do it. When I talked about the threat to the indigenous people up in Alaska, I wanted to point out that the saxitoxin that's used there is a very potent toxin, and it goes back to the days that this group would remember Francis Gary Powers. We all were of that generation, the U-2 pilot that was shot down over Russia. Well, he carried a silver dollar that had in it a little needle that was coated with saxitoxin. That was what he was supposed to take, but never did. And they had to do a prisoner exchange. But a lot of the early work on saxitoxin was funded by the Army, actually, so that they could make these, what could be then these very potent toxins. So they're, when dried, they're still potent. It is a risk. Same for marine, same for fresh water. So be careful with the fertilizer idea. But on the other hand, one of the problems you have along the coast of Florida are seaweed blooms of sargassum and alga and others, and they don't produce these toxins. And that can be composted, that can be turned into fertilizer readily and would have no harm at all.
Okay, but Don, there's another group doing the sargassum as well, they're harvesting it for other chemicals. But the reason we're not interested in sargassum is it's loaded with arsenic. Yeah. And arsenic is a small molecule, and it will go in the roots. It will end up in the strawberries. And to my knowledge, arsenic also causes cancer. It's a neurotoxin. It's not something you want to load your body up with. so that rules out that. I mean, Sharon is trying to solve two problems at once. She will dramatically reduce the phosphorus in the Lake Okeechobee and fertilizer and biological mass. And but we were going to put it down as not as a sprayed fertilizer, but as a sort of a solid moss. We can't afford to ship water around, so it's going to be dewatered but still wet and be put down as a solid fertilizer, probably buried an inch or two under the soil automatically by the tractor that puts the straw is highly automated.
Yeah,
And
that would be that. See, what I was talking about, we're spraying the water over the plants where it then dries and remains on the surfaces. But if you're putting it into the soil, there are organisms that'll break these toxins down fairly quickly into smaller components that are then either the fertilizers or just whatever's needed by these plants. So I would worry less if you can actually get it into the soil.
Don, you're a fabulous speaker. This was fascinating. Who knew? I didn't. And thank you so much. Don, do you want to say a final few words on anything?
Just what I'm pleased, I see a number of friends, and I can't say hello to all of you. I'm glad that a number of you joined, but I'm also glad that a lot of people that I don't know joined. And I hope when we eventually get together, either as a class of 70 or a larger group, that now people will know who I am. Happy to meet the rest of you in person. I hope that comes soon.